Buddy Ratner from University of Washington. He is the director of University of Washington Engineered Biomaterials, co-director of the Center for Dialysis Innovation, and the Darlon Endo Chair in Technology Commercialization. He's a professor of bioengineering and chemical engineering at UW, got his PhD in 1972 in polymer chemistry from the Polytechnic Institute of Brooklyn. He is a fellow of many institutes, including American Institute of Medical and Biological Engineering, AVS, AAAS, ACS, ACS Poly, and International College of Fellows, Biomaterial Science and Engineering. In 2002, Ratner was elected to the National Academy of Engineering. He has launched 7 companies and won numerous awards, including AVS Welch Award 2002, Society of Biomaterials Founders Award, BMES Pritzker Distinguished Lecturer, Acto Biomaterialia Gold Award, Galetti Award, among the others. He has served as president of SFB, Society for Biomaterials, and president of AIMBE. He has led the most well-known textbook in the materials area, Biomaterial Science and Introduction to Materials in Medicine, which has sold over 40,000 copies and used in most biomaterials education programs worldwide. The floor is yours, sir.
Okay, Mehmet, thank you very much for that introduction. Thanks for the opportunity to share with the Terasaki Institute some ideas that I think are, let's see, how to put this well. Kidney dialysis is maybe these days not been a mainstream subject. And I'm going to try and change your mind about that particular issue. So I call it Rethinking Kidney Dialysis, and I have past, present, and future there. And we will be shuttling around in time quite a bit in this talk. So in fact, these are just some of the years that we're going to visit in trying to put this story together. And maybe the most important thing is 2021 plus. So I just emphasize my primary interests for a long time have been in the actual medical devices, and especially the biomaterials that we make medical devices of. And this talk is going to focus on this particular medical device, a kidney dialysis or hemodialysis. So let me start here. That's a photograph of a dialysis center in Hawaii, in Maui. And there are multiple dialysis centers in every city and town. The dialysis centers literally sustain the lives of maybe 3 to 4 million people worldwide. And without these centers, I think there'd be much less chance for their survival. So if this is such a major enterprise worldwide, dialysis centers, why do we need research on dialysis? And maybe we say, is dialysis a solved problem already? When I started my career in the 1970s, there was a lot of research going on dialysis, and I don't see that much today. So maybe it's an old-fashioned theme. Well, let me see if I can change your minds about these questions. I believe dialysis is not a solved problem, and it is not an old-fashioned theme. But speaking of old-fashioned, let me start here with one of the great pioneers in medical devices and biomaterials. This is Dr. Wilhelm Kolff. And Dr. Kolff was a young practicing physician in a small town in Holland during the Nazi-occupied Holland. And when a patient was brought to him with kidney failure, Dr. Kolff was distressed because he knew he could do nothing for those patients, and he could not give any reassurance to the patients or the families, and it greatly distressed him to see his patients just dying, and he couldn't do anything for them. He came to appreciate that the issue was a buildup of toxins in their blood, and started experimenting with just dialysis membranes. He put some blood in a dialysis membrane and noted that the urea quickly diffused out of the blood into the dialysate, just basically saline solution. And to actually read Dr. Kolf's words, I decided that if I could remove from this patient just as much urea as he formed every day, that he could live. That was actually quite a brilliant insight that Dr. Kolf had, and set him on a path to invent kidney dialysis. So Dr. Kolf, again, in a very difficult situation, in this Nazi-occupied Holland situation, built the first dialysis machine. He used things like a Ford water pump, washing machine parts, orange juice cans, sausage casings, put together a whole bunch of equipment to build this first dialysis system. And in that era, he could do, well, he had many patients coming to him. When a patient went into a uremic coma, coma caused by kidney failure and buildup of those toxins, no patient had ever recovered, come out of that coma. It was the path to death. And so he felt justified in exploring, testing his dialysis device out on such patients. And he did have a number of, let's call it failures, but these patients were essentially doomed anyway. Finally, this lady at the bottom, you see her picture there, Sophie Schaffstadt, succumbed to one of these uremic comas and was put on this machine. And many, many hours later, she began to open her eyes and began to speak. And so she is probably the first human in history, the history of the human race, to come out from a uremic coma. And she opened her eyes, and her first historic words were, "I'm going to divorce my husband." Now, that's a pretty amazing story, but I heard Dr. Kolf tell it himself, so I think there might be some veracity to it. Okay, I'm going to jump forward a bit. Let me just mention that Dr. Kolf's development, this kidney dialysis system, was brilliant and could bring patients out of this coma. But of course, the patient then built up uremic toxins again and had to be reconnected to that machine. And it required a major surgical intervention every time you connected a patient to the machine. And eventually the patient simply ran out of sites that they could hook the patient up to. And so it was not suitable for long-term or chronic dialysis. It could get a patient out of that condition, but could not keep a patient chronically on dialysis. So that's why I'm going to jump forward to 1960, where the next major innovation happened in kidney dialysis. And just to put 1960 in context, 1960, the word biomaterial had not been yet used in the literature. The word biomaterial didn't exist. The word biocompatibility didn't exist in 1960. There was no medical device regulation. There was almost essentially no medical device industry. It was practically a cottage industry. And really, there was only one society that sort of addressed medical devices and biomaterials, and that was ASIO, the American Society for Artificial Internal Organs. So it was a very different era from today, where we so appreciate medical devices, biocompatibility, biomaterials. That's 1960. Well, I'm showing a picture of three individuals. The picture was actually taken in 1992, but these three individuals did some amazing work about 1960. So in the center, we see Belding Scribner. an MD nephrologist, a visionary. And on one side is Les Babb, a chemical engineer. On the other side is Wayne Quinton, a bioengineer. And these three people put together built the first kidney dialysis systems as we know them today. And as I say at the bottom of this slide, this happened at the University of Washington or the University of Washington Medical School. So this University of Washington collaboration between physicians and engineers led to this therapy that is now sustaining the life of 4 million people worldwide every year. And one of the great components of this innovation was what's called the Quinton-Scribner shunt. And this allowed access to the patient's blood without requiring a major surgical intervention each time, allowed a patient to come to a dialysis center, be plugged in, and be dialyzed. The shunt was based upon a brand new material that Wayne Quinton had learned about that was very exciting to him in that maybe 1958, 1960 era. That material was called Teflon. It was just released and it seemed to have amazing properties. And Wayne Quinton used it as the blood access component of the shunt device. So here we see in 1960, Clyde Shields, the first human who was kept alive on chronic dialysis. We're looking at Clyde in bed. Just keep in mind that every single person before him whose kidney failed died. So Clyde Shields actually was on this chronic dialysis about 20, 25 years. So that was a great extension of his life due to this therapy. And you can see the blood coming out of his arm. Again, he's using this Quinton-Scribner shunt and basically going into dialysis membrane and coming back to his body. We'll talk a little more about the procedure shortly. Dr. Scribner appreciated that many patients are going to need this and that the hospital setting maybe isn't the best place to give this chronic dialysis. So he launched the Northwest Kidney Center, the world's first dialysis center. And again, this allowed people to come in, be dialyzed, go home. So that is kind of the launch of chronic dialysis as we know it today. And 60 years have passed since these incredibly visionary, pioneering efforts. Have things changed much? Let me add one other point here. I guess I'm just interposing this. This is my PhD dissertation in 1972, and I was working all the way back in you might call it the early days of this on the interaction of urea with polyhema hydrogel membranes. Thesis was mostly about transport to the membrane, and again, research in this area was quite exciting and active back in that era. So anyway. Kidney dialysis, we're going up to maybe 2000. By 2000, the dialysis machine, as we know it today, was pretty much developed, finalized, established. And we have on the left side of this image, basically what's going on today. We take the blood out of the patient's arm, put it through a hollow fiber dialyzer, dialyze it, the blood against a large volume of water, return the blood to the patient. So dialysis, really, this is basically what Belting Scribner did with his patient, Clyde Shields, back in 1960. The machines now have beautiful digital displays and all. Didn't have anything like that back then. But still, basically, the same pattern that we see in the left side of this image going on today. So in the first decade of the 21st century, we spent already about, so that would be 2000 to maybe 2010, we spent greater than $2 trillion on dialysis. And every 10 years, we spend worldwide that sort of sum of money. So what are the issues and concerns? You know, we're doing it, again, on millions of people. And we will address each of these, but the talk will be focused on the technology and medical issues. But we're going to talk a little bit about access and disparity issues, about the economics of kidney dialysis, about environmental issues, and then go on and focus on technology and medicine. So the concerns for kidney dialysis today, when a patient starts kidney dialysis, average lifespans are 4 years. It is very hard on patients, and many people simply can't deal with this very difficult therapy. Dialysis patients are, as I like to say, rarely happy. They're tied to a heavy machine 3 times a week for 4-hour sessions, and then they don't feel very good the rest of the time. They symptoms include nausea, itching, body pains, exhaustion, cramps. There's a whole list of symptoms, but basically it is quite a debilitating therapy. The costs to the world, as I've said, are huge, certainly greater than $130 billion a year. And incidentally, 1% of the total US budget goes to kidney dialysis. Now, that's not the healthcare budget. 1% of the total US budget goes to sustaining dialysis patients. There's been a little innovation in the whole field since that sort of pioneering work in the 1960s in Seattle. And each year, this is from an article in The Lancet, about 7.1 million people worldwide are literally sentenced to death because of lack of access to kidney dialysis. It is simply for low resource environments, simply too complex, too expensive, and people whose kidneys fail are simply told you're going to die. So that is part of the incredible disparity that we have in the way we administer this kidney dialysis therapy. So there are many technical issues. The technical issues are listed on the left, and I like to think we have biomaterial solutions that we can bring to the table in the 21st century. So we're talking about a 20th century technology. It's been around 60, 70 years. And can we bring exciting new technologies to bear on this? So the technology medical issues include things like blood access, thrombosis, infection, membrane fouling, incomplete waste removal. And I will go through most of these, not quite all of them, and show you how we can make progress there. I mentioned environmental impact, and environmental impact is simply not sustainable for what we do in kidney dialysis. Water used is a tremendous amount of very high quality water per year, and many environments have serious water shortages. Electricity consumed is huge, and waste produced are gigantic in this dialysis treatment. For example, the polyvinyl chloride tubing, PVC tubing, used in dialysis sets takes about 20 feet of dialysis tubing per treatment. And you multiply that by the 4 million people worldwide, each getting 3 treatments a week for 52 days a year. And you come up with a length of tubing that's enough to go from the moon and back 3.5 times. And we take all that tubing and just chuck it into the waste. So that too needs addressing. So again, specific biomaterials issues with kidney dialysis, the blood access situation, closely related to that are blood vessel hyperplasia and thrombosis, infection, membrane fouling, incomplete waste removal. Again, we're going to address most of these points. But many people say, hey, why don't we use tissue engineering to address the kidney dialysis crisis? And I'm personally quite involved in tissue engineering. I do believe it is the future. I think it will be the future for kidney dialysis. Huge potential to it. But another way to look at it a bit more cynically is that it's been about 30 years since the first important papers on tissue engineering came out. And if one looks at the impact that tissue engineering in 30-something years has had on medicine, I'd say the contribution to day-to-day clinical medicine has been nearly negligible. There's a few small examples, but mostly tissue engineering has not made the contribution we think. And so we concluded that to get a therapy to patients quickly, a mechanical biomaterial strategy will be much faster. And that's the approach we're embracing here. We have a predicate for portable dialysis innovation. Look at the left ventricular assist device, the so-called artificial heart. In 1993, the artificial heart involved a huge electronics console to power the whole system. By 2007, all this was reduced to sort of a rollerboard-sized device. And today, you can see hanging on the waist of this individual is the complete power pack. So modern technology has allowed great reduction in the size and complexity of medical devices. And we're using this as sort of the predicate for developing portable dialysis. So let's talk about a wearable artificial kidney. Again, historically, we can look at literature and find patents and papers going back to at least 1965 envisioned wearable artificial kidneys. The idea has been here a long time. So you've asked the question, why today do we still not have a wearable artificial kidney? Well, technologically, this is extremely difficult to do. But there was a clinical trial on a wearable artificial kidney. In this picture, we see in the center is Victor Gura, an inventor who invented a wearable kidney. To the left is Professor Jonathan Himmelfarb. Jonathan, you'll hear more about, is my colleague and co-director of our dialysis center. Center for Dialysis Innovation, and to the right is Professor Larry Kessler from our School of Public Health. And these three individuals did artificial kidney. So you can sort of see where in this device is quite thrilled. Actually, in the left side image, her mom is in bed on a conventional dialysis machine. So that all looks great, except the trial didn't go well. It was discontinued often after 8 hours with concerns for patient safety. And so this was an important first step, but it did highlight the complexity and problems of doing, and stimulate us to launch the Center for Dialysis Innovation at the University of Washington. As I mentioned, I co-directed with Professor Jonathan Himmelfarb, a MD nephrologist. And one of the first things we did was bring in a patient advisory group. We had to find out what patients really wanted and what they're going through. And we took down a number of quotations. This from one patient: "This is ridiculous. We need to find a better way." And it really became almost our mantra. So we had a launch event. You can see the tall gentleman to the right there is our Dean of Medicine, Paul Ramsey. And Dr. Himmelfarb was making an announcement, and we're launching this center 2017. We have actually, I think, a very appropriate leadership group to do this. Again, Jonathan, expert in medical issues associated with kidney dialysis, myself, a bioengineer, Dr. Kessler, who I introduced earlier, worked about 15 years at the FDA, headed one of the divisions. Dr. Thompson is in charge of sort of IP and translation and commercialization. Glenda Roberts was on dialysis as a kidney transplant patient now, and she is our Director of External Relations and Patient Engagement. Dr. Galperin is a PhD polymer chemist, worked in industry for a number of years and as our director of research and development. And Barry Fulkerson is our director of systems engineering, and Barry has been involved in the development of almost every kidney dialysis machine that's on the market today. Very experienced in how dialysis machines are developed. So one of our other important thinkings are your kidneys work 24/7. And how can we expect 3 4-hour dialysis treatments a week to emulate the natural kidney? When you think about it, if you're going to get 3 4-hour treatments a week, your blood toxins build up to high levels. You get dialyzed, they crash down, build up to high levels, crash down over and over again. Your kidneys keep your blood toxins at a very low constant level. And so that's physiologically normal. And maybe many of the complications from dialysis as we do it today are associated with this infrequent dialysis. So our original, we call it sort of our aspirational thinking, was maybe a wearable dialysis system. There might be some little power pack that runs the whole thing. This was really pretty aspirational. But we did name the device, we call it the ambulatory kidney to improve dialysis. And today we're thinking about a generation 1 that's really going to be a portable, a rollerboard. And then the generation 2, we do hope to really take this to a wearable form factor. So what do we need here? Portable, wearable, miniaturized, water efficient, environmentally more friendly, minimum coagulation for the patient, robust design, cost effective, patient centered, and ultimately should restore kidney health. Our Center for Dialysis Innovation set up a timeline. If we looked at 2017, when we started, all dialysis is associated with those big machines shown in the upper left. In fact, since 1960, they were associated with it. That was our day 1 of our CDI. And we gave ourselves a 5-year timeline to reach an actual clinical trial on this. I must say, the COVID situation put somewhat a crimp on our timeline, but we think by maybe 2022, 2023, we actually can get some of our developments to patients. So what do we need for a wearable kidney? Well, ultimately, we have to remove about 15 grams of urea a day. That's what your body makes. Some other toxins have to be removed. Now, if we have a wearable kidney, the amount of water we can use in this should be 1 kilogram or less. We'll talk about water in a minute. The total weight must be really less than about 12 kilograms. Even that's kind of heavy for a patient. We have to control electrolyte balance, fluid management. There has to be modest power consumption, patient safety up front, onboard monitoring, control, robust and reliable, patient-centric, user-friendly, maybe even affordable. These are all things we're thinking about. So I mentioned water. Kidney dialysis, as we said, is very water consumptive. Every dialysis center has a water purification system. You can see one of these in the body. They're complicated and expensive to give the high quality water we need for dialysis. About 400 liters of water is converted to 120 kilograms, 120 liters of very high quality water. For our wearable and portable dialysis, we want to do that with 1 kilogram of water. So how do we do that? Well, let me go into some of the details here. So we see a number of components that seamlessly integrate for the active kidney device, the wearable and portable dialysis we're envisioning. And you can see each of these components, removing toxins, improved membranes, sensors, addressing infection, bacteria and biofilms, blood access, blood compatibility, device design. All these have to fit seamlessly to make our kidney dialysis system work. And so we're going to examine sort of each piece of the puzzle here. So let's talk about removal of toxins, and closely connected with that is improved membranes. See some of the investigators. We have the Center for Dialysis Innovation is about 10 professors, about 30 students and researchers. And the challenges, again, I mentioned we have to remove 15 grams of urea a day, and membranes lose their efficiency. We want to use a small quantity of water. There are other toxins that have to be removed. We have to measure and analyze these toxins, and also ultimately address electrolyte balance. So one of the remarkable innovations comes from one of our CDI investigators, Professor Bruce Hines in our University of Washington Materials Science and Engineering Department. And Bruce developed titanium dioxide nanowires that can be used in a photocatalytic system. And he demonstrated that we can take urea, pass it over these nanowires in the presence of LED light, and convert that urea to CO2 and nitrogen. So instead of dialyzing against a large quantity of water, now we can dialyze against a smaller volume of water. So this sort of artist's illustration shows really what's going on. Off to the left is the patient. The patient's blood is going through that hollow fiber dialyzer. The dialysate comes in. It picks up the urea from the blood. It goes into the photooxidation unit, which you see on the right, is passed over the catalyst in the presence of UV light, and the urea goes off as nitrogen CO2, and then that dialysate can go back into the hollow fiber dialyzer and pick up more urea. So that's the system we envision. How does it work? Well, here's some real data. On the left, you can see the experiment. What we're doing is continuously infusing urea into a tank at about the same rate your body makes urea a day. This would lead to about 15 grams of urea added into the tank during the day. And with the lights out on the DRM, the dialysis regeneration module, you can see the red curve, the urea concentration is steadily going up in the tank. Why? Because we're adding urea. When we turn the digital regeneration module lights on, see the blue curve, the urea concentration of the tank is remaining relatively quite steady. Again, that urea is being turned into nitrogen and CO2. On the right, we're looking at some of the other toxins that also have to be removed. And interestingly, this UV photocatalytic system pretty much burns up any organic, and so that has a number of advantages. We know for this whole system, that's how we're going to get rid of urea. We know for this whole system, we also need improved biomaterials. Again, a patient wearing this device long-term, we can't be replacing the tubing every 4 hours, can't be replacing the membranes, and the patient can't be continuously anticoagulated. That patient, we have to have tubing, membranes, blood components that are much more resistant to fouling and to blood elements. And one of the innovations that may help us get there was developed by another one of our CDI investigators, Professor Xiaoyi Zhang in our chemical engineering department. And he developed a class of polymers called Carboxy Betaine Methacrylate polymers. These are sphitter ionic polymers, and in quite a large number of publications, they've been shown to be super low fouling, even at solutions of full physiologic concentrations. So still another issue here is that hemodialysis patients are exposed to all this PVC tubing, and they tend to have high levels of plasticizer in their bloodstream. These plasticizers in PVC have been shown to be hormonal disruptors, endocrine disruptors, and are a great concern in medicine at the present time. So can we reduce plasticize and leaching. So there's the structure of a polymer we developed specifically to treat dialysis components. In that chemical structure on the left, you can see the plus minus of the zwitterionic structure, the carboxy betaine. On the right, there's a photoreactive group, a benzophenone type group that allows us to photoreact the zwitterionic polymer in a very facile manner with most materials and surfaces. And so we treated PVC tubing with this zwitterionic polymer. And here's just some of the data. I'll show you a little more later in this talk. But the orange bars in this are the uncoated or controlled PVC. The blue are coated. And if we look at again, I mentioned we could reduce protein fouling. You can see that our polycarboxane coated materials, the blue, have much lower protein fouling. That's off to the left. Off to the right, we look at plasticizer leaching. And again, the polycarboxy betaine methacrylate, the coated, the blue, greatly reduced the plasticizer leaching. We published this work in ACS Applied Materials and Interfaces. So we're actually treating along with the PVC tubing intact hollow fiber dialyzers with this particular types of coating. Let's talk about another challenge, blood access. So the vascular access grafts we use in dialysis today fail, and I'll talk a little bit more. Actually, these comments talk to how we do blood access today. It's different from the Quentin Scribner shunt done in the 1960s. These days, we use these big needles. These are stuck in the patient's arm 3 times a week, 2 of those big needles. Our patient advisory group tells us that's probably the thing they hate the most about the dialysis treatment they're getting is these big needle sticks. And these needle sticks are disfiguring too. So there's really 3 modes we use to access patients' blood for dialysis, all of them serious problems. We're going to talk about the central one, which is an arteriovenous, an AV graft. And the 2 needles are stuck into the synthetic graft between the artery and the vein. These have a 50% failure rate at 1 year, huge failure rate. And as you can see from the image of the arm, they're quite due to the needle sticks, they're quite disfiguring to the arms also. So our rationale for the Center for Dialysis Innovation Vascular Graft Development is we feel that vascular grafts' 50% failure rate is due to chronic inflammation. It really is quite evident the body never stops attacking them. So we address inflammation. And we developed a class of materials in our group at the University of Washington. We call them 6S. I won't go into the details, but these are materials where all pores are 40 microns in diameter. Pores are interconnected. And we found in many experiments that they mitigate chronic inflammation. How do they do that? Well, this is an artist's representation, but it turns out that macrophages heavily invade these, and yet the macrophages get shifted to a pro-regenerative phenotype. We've published a number of papers on this aspect. So for a vascular prosthesis, a vascular access graft, what properties do we need? We need blood compatibility. That might come from endothelialization. We need a biomechanics to match that of the blood vessel. It should be suturable. It should be durable. And one other aspect is we've come to the appreciation that your natural arteries actually have their own blood vessel network. It's called the vasa vasorum. And can we build into a synthetic active vasa vasorum network? So this is the vascular graft we've imagined. It uses our 6S porous material. You can see an artist's conception of what it looks like on the bottom. It's lined with gelatin. The gelatin we found degrades in just a few days, but it does seal this porous material against blood leakage. And what we hope to do, if you look at the upper right cartoon, is that from the tissue side of the vessel, we want blood vessels to ingrow and literally seed or nucleate the endothelial layer on the lumen of this device. To do this, we invented a new polyurethane. PhD student Lejeune and Dr. Felix Simonovsky, by the way, Lejeune is now Dr. Jeune, got his PhD doing this work, invented a new polyurethane that uses some conventional components, but also uses some very unique cross-linkers that lead to a very biostable polyurethane. And one of the good things about these polyurethanes is we can adjust the components, the ratios of components to go into them to adjust the mechanics of the material. And on the y-axis, we have Young's modulus. And by adjusting the soft segment, hard segment ratios, we can tune the mechanics of these polyurethanes to that of native artery, which is what you see here. So this is just some SEMs showing the wall structure and what this material actually looks like. And Lejeune made this unique implant. The left side is a solid material of this polyurethane. The right side is this unique porous structure. It was implanted for a month, in this case subcutaneously. And you can see on the left side, if you look at the histology image, you can see the dense blue line from trichrome staining that's indicative of a dense foreign body capsule. Switch to the right side of the histology, lower right side of the histology image, you can see there's almost no foreign body capsule, and the structure is heavily infused with blood vessels. We can blow up those blood vessels, see the blood vessel structures, and stain for the blood vessels. And this is our pseudo-vasovasorum. We did long-term sheep implants on these, and to the right, you can see the control, the conventional porous Teflon graft that's used today. Dr. Eleni Quiroga is a highly experienced vascular access surgeon. She puts these in all day long in humans. She put these into sheep. And upon harvest, she's pointing out to us the dense foreign body capsule, the dense scar layer that forms over these. And she says, this is what we see in humans when we put these in. When she opened the implant site for the 6S, or the new graft we developed, her comments are very interesting. She opened it up and looked at it and said, well, there's something wrong here. I know I implanted the device here, but all I see is a living, beating blood vessel. Where did my graft go? And well, I must say that's exactly what we were trying to do. So it's very exciting. We're still working on the histology, but the initial analysis shows cellularization occurring in the 6S material. There's a lot more thrombotic material in the porous Teflon on the bottom. And this development, this vascular graft development, won a $500,000 Kidney X prize from HHS and the American Society for Nephrology. We're also trying to 3D print it these days using a very high resolution 3D printer. And we have sort of models of what this might look like if it was 3D printed. Blood compatibility gives us a whole bunch more challenges. I'm going to sort of skip the words and get a little short on time here, and I want to get to some of this. So we have a rationale. We've been working on blood compatibility since the 1970s, and our rationale is two components. One is using albumin-attracting biomaterials and protein-resistant biomaterials. The albumin attraction hypothesis goes all the way back to the 1970s, and Professor Don Lyman really was one of the pioneers in introducing this idea of increasing the concentration of albumin on the surface of things in the bloodstream. Dr. Lyman recently passed on. We've lost a great pioneer there. But we've invented certain plasma deposited, that's radio frequency glow discharge, plasma deposited fluoropolymers Two of them are circled on the bottom. And what we're looking is the percent albumin retained. You can see we have materials that retain from the bloodstream a very high level of albumin. And that's good. That's what we want to see. So we've done many blood compatibility assays or blood interaction assays. No one assay really gives the whole story. But circled in the blue boxes are both the C3F6 plasma deposited fluoropolymer and the carboxy betaine. You can see in every single case, the C3F6 polymer and the carboxy betaine polymer give improved results in blood compatibility. So we're starting to introduce these into our dialysis system. So again, these are the conclusions I just gave you. We also have challenges. We need to continually sense what's going on and measure it. So a dialysis nurse won't be with the patient 24/7. We have to control the active to ensure patient safety, and we need sensors for another component. This is another aspect of work that's ongoing now. Bacteria and biofilms, we have to inhibit those, particularly at the skin interface site for things we're developing. And actually, something like 10% of dialysis patients today die of sepsis. So we're working on other strategies for that. I'm not going to go into that. And then there's the whole issue of device design and engineering. And so we have a team working on that to get components working together, low power requirement, low weight, safety. We have patents, regulatory and reimbursement issues, and manufacturing and selling. So we think the ultimate challenge really is how to get a university academic development to patients. And so we're very intent on commercializing our technologies, and companies can convert technology into something that can pass through the FDA with appropriate design controls, ISO standards. And so we spun off a company. We call it Kuleana Technology. And Kuleana is a Hawaiian that says work. It should be Hawaiian word, meaning responsibility and care. It's the definition in the Hawaiian language of Kuleana. And we think that defines what we're going with this program. So we're developing what we think is a very novel university industry partnership where CDI becomes the skunk works for developing technologies. We can do that on much more economical basis. And we have the talent at the university to do this research. And then we pass things over to Kuleana, the development company, which can deal with the FDA, can deal with licensing, can deal with reimbursement issues, and raise funds to do all this. I keep on emphasizing that the patient voice, we have involved in every aspect of our development. We have a large patient advisory group and many different panels, and the patient voice, the patients have to contribute to get a device that they will wear and they will embrace. So in conclusion, this is a nice photo of Belting Scribner. He's passed away a number of years ago, but he did say if the treatment of chronic uremia cannot fully rehabilitate the patient, the treatment is inadequate. Scribner said that in 1963. I think that's still the case, and we're trying to change that and to fulfill Belting Scribner's vision. So sort of a recap, my career was launched basically with research starting in '67 with experiments relevant to kidney dialysis. And as I say, here we go again. We're going to quickly translate biomedical ideas to patients. It's one of our missions, patient-centric. And we still have these profound problems in hemodialysis 60 years after developing systems. So it's just a nice photo of my group in an era where we could all be together without mess and kind of be happy. But I still have a nice group, and we're all working actively on kidney dialysis. So I'm going to thank you very much for your attention. I'm going to stop the slide sharing, and I'd be glad to take questions. Thank you.
Thank you for the fantastic talk, Professor Ratner. A couple questions came in. One is it plausible to develop a wearable system that can be used between patients? Would that increase the cost since you have to integrate more disposable into the system?
Could you repeat the starting of the question? I'm sorry, I missed the very...
Is it possible to develop a wearable system that can be used between patients?
Between patients. You know, we do see that this wearable system would be dedicated to one patient. It really simplifies a lot of issues, but we are envisioning, or let's call it versions of our system that would go into conventional dialysis centers today. And those would have to go between patient to patient. So we are addressing the issues of how we're going to sterilize and what the parts must be replaced and what parts could be used over and over again between patients. So we are really addressing that issue. Yet the wearable, we do envision that each patient would have their own wearable device.
Thank you. Another person says, excellent presentation, Professor Ratner. He says, the 6S biomaterial for vascular access seems very interesting. There's some research that is suggesting nanofibers as an optimal structure for vascular access, as it allows cell ingrowth epithelialization, mimicking a native vessel with the added protection that these cells provide against thrombosis and infection, as well as the benefit of early puncture cannulation. What are the advantages of the 6S graft compared to electrospun nanofibrous grafts? Do you see other opportunities for nanofibers as a solution for dialysis, like filtration membranes?
Well, we are personally not working in nanofibers. I've actually published a number of papers in that area too. It's a very interesting class of materials, but we have explored pore sizes, range of pore sizes to come up with the 6S material that is 40 micron pore size. And we found that smaller and larger pore sizes simply give the conventional foreign body reaction with the fibrotic reaction. So we have embraced this 40 micron pore size as being ideal. And it's actually gone into many different sites in the body. It heals subcutaneously, it heals percutaneously, it heals into heart muscle without scar. And yet when we use other pore sizes, we do not get this good result. So back to nanofibrous materials, they tend to have very small pore sizes, very irregular pore size distributions, not uniform distributions. And so I believe most nanofibrous materials, if they're simply implanted as a biomaterial, give the conventional foreign body reaction, which is a nonstop inflammatory reaction. It may meet all the FDA criteria of biocompatibility, but they give this fibrotic, inflammatory foreign body reaction. And we're trying to get away from that reaction. So that's the rationale for the material. And that's why we're actually not working in these nanofibrous materials at this present time.
Thank you. What is the most significant limitation for a wearable artificial kidney, the size or efficacy?
Well, I think it's getting all these different components. And again, there are biological components, performance components, mechanical components, safety components, getting them all to work together in a reliable manner such that we can send somebody out into the world. Again, people have done things like this with the left ventricular assist device, send patients out into the world with an implanted device of that complexity. We think we can do it with a kidney too. But there's a lot of moving parts, if you will, in such a device, and we have as the wearable artificial kidney, and we are trying to think through how we can do that, and hopefully addressing each of the components.
Thank you. How do you prevent blood clot formation in the wearable artificial kidney?
Well, again, the issue is much better blood compatible materials. As I said, I started my research on blood compatible materials really in 1972 was really the first efforts I started in that area. And I could say right up today, there's no list of what is a blood compatible material. There are no standards for what is a blood compatible material. There are a lot of things claimed to be blood compatible. I think there are a lot of materials that do show improved blood compatibility, but we need much better blood compatibility. We can't keep a patient heparinized 24/7. At the moment, they're put on a conventional dialysis machine and they just give them a dose of heparin so they don't clot. Without the dose of heparin, there'd be tremendous clotting problems. And so we are going back, rethinking, again, 60, 70, 80 years of research on blood compatible materials. And we think we have some defensive hypotheses and some materials that look quite exceptional. And so the zwitterionic polymer, the fluoropolymers work particularly well in high blood shear environments. The zwitterionic polymers seem to work in most shear flow or shear environments. And so we're embracing those materials.
Thank you. About macrophage activation, can the needles be coated with an anti-inflammatory drug to prevent macrophage activation?
Needles could be done with that. The classic anti-inflammatory drugs are steroids. And one of the issues we have with those, yes, they'll turn off inflammation, but they also turn off any healing or regeneration. So another project I didn't talk about here is we're trying to get a skin interface that literally heals to the skin. We don't want to just stop inflammation. We want to encourage healing. And also for our implanted vascular access grafts. Again, we want healing, not to just stop inflammation. It turns out inflammation has a bad reputation, but the medical textbooks define inflammation as the normal process of healing in vascularized tissue. And we want to promote that normal healing. It is our biocompatible biomaterials that give abnormal inflammation and aberrant healing.
Thank you. Urea is one of the vital components to remove. How about other compounds such as lactic acid? Is that a problem?
Lactic acid is not a problem. Your body has very excellent mechanisms for clearing lactic acid. But there are other components, creatinine, and there's a number of other toxins with names like indoxyl sulfate, paracresyl sulfate, a few other of these toxins that many people are responsible for many of the cardiovascular complications that go hand in hand with kidney dialysis treatment. And we are actively working on strategies to address and get rid of, along with urea, these other toxins.
Thank you. Another question came in. How do you intend to inhibit fibroblasts?
Inhibit fibroblasts?
Yes.
Yeah. Well, again, in our studies of the foreign body reaction with this so-called 6S material, the material with interconnected 40 micron pore sizes, the fibroblasts, yes, are present. We see fibroblast-like cells in the interstices, but they do not seem to be in the mode where they create or give off this collagen and make this dense collagen layer. So fibroblasts are, I think, a part of the normal tissue, but they can also be programmed or directed to make this foreign body capsule, the scar. And in our case, they do not seem to be turned on to do that process.
Another person says, do you have any plan addressing lactic acidosis issue, which is very common in critical care units as well?
Again, we have not identified that as a particular problem in patients on chronic dialysis. And again, we're very in tune with the clinical or medical issues associated here.
Thank you. Is there any chance of infection, like bacterial infection, for long-term application? Can we coat any antibacterial polymer which can increase biocompatibility with blood and decrease infection as well?
Yeah, so the sphincterionic carboxy betaine polymer has shown an incredible ability to resist bacterial biofilms and bacterial adhesion. And that's going to be one of our major strategies for preventing biofilms and bacterial adhesion in the systems we are developing.
Thank you. Well, that's all the questions I have. Thank you for the great talk, history, everything, sir. I appreciate your time.
Okay, it's been a pleasure communicating with people, and I'm easy to contact by email. People have further questions or interested in our program, or look at our website, the Center for Dialysis Innovation. Thank you for your attention.
Thank you, sir. Have a nice day.